Tonight's lecture is over Chapter 4, Cells. Our learning objectives for this lecture include defining the cell and being able to distinguish between the two general types of cells. We will also be able to describe the structure and functions of cell membranes. We will be able to describe several ways in which molecules move across membranes. We will also be able to describe how cells are connected and how they communicate with one another. And finally, we will describe 9 important landmarks in eukaryotic cells. Whether we are studying a creature as small as a flea or as large as an elephant, all organisms are made of smaller units that are more easily studied and understood. The most basic unit of any organism is the cell, the smallest unit of life that can function independently and perform all the necessary functions of life. The term cell was first used in the mid-17th century by Robert Hooke, an English scientist who thought the magnified views of a very thin piece of cork resembled a mass of empty rooms. He named these compartments cellulae, Latin for small rooms. By the 1830s, improvements in microscopes helped scientists realize that all plants and animals were made entirely from cells. Subsequent studies revealed that every cell seemed to arise from the division of another cell. For example, your body is made up of about 30 trillion cells, all of which came from the single fertilized egg produced when an egg cell from your mother was fertilized by a sperm cell from your father. Moving on, the facts that all living organisms are made up of one or more cells and that all cells arise from other pre-existing living cells are the foundations of what we call cell theory. Cell theory is one of the unifying theories in biology and one that is universally accepted by all biologists. The origin of life on Earth was a one-time deviation from cell theory. The first cells probably originated from free-floating molecules in Earth's ancient oceans, at least 3.5 billion years ago. Since that time, however, all cells, and thus all life, have been produced as a continuous line of cells originating from these initial cells. Today, we know that the cell is a three-dimensional structure, like a fluid-filled balloon, in which many of the essential chemical reactions of life take place. Generally, these reactions involve transporting raw materials and fuel into the cell and exporting finished materials and waste products out of the cell. In addition, most, but not all cell types contain DNA. This molecule, which we learned about in a previous chapter, contains the information that directs the formation of various cellular products within the cell, the chemical reactions in the cell, and the cell's ability to reproduce itself. Nearly all cells, even in multicellular organisms, contain the genetic information for the entire organism. And the key words are nearly all cells, meaning not all cells contain the genetic information for the entire organism. For example, in humans, red blood cells do not contain DNA. Furthermore, the sperm and the egg contain only half of the organism's genetic information, making them haploid cells. Moving on, most cells are too small to see with the naked eye. For instance, at this very moment, even if you brushed your teeth, there are probably more than 7 billion bacterial cells in your mouth. But I should point out that there are a few exceptions to this. For instance, there are some oversized cells, including some squid neurons that can be many meters long but very thin. And by volume, the ostrich egg, which weighs about 3 pounds, probably contains the largest of all animal cells. Eggs also tend to be the most valuable cells. Almus caviar, which consists of eggs from the beluga sturgeon, these cells sell for nearly $700 per ounce. Human eggs can also be sold for thousands of dollars per egg on the open market. And that brings us to our take-home message for section 4.1. The most basic unit of any organism is the cell, the smallest unit of life that can function independently and perform all of the necessary tasks of life. This includes reproducing itself. All organisms, or I should say all living organisms, are made up of one or more cells, and all cells arise from other pre-existing cells. Moving on. Although millions of diverse species live on Earth, and many of those organisms contain trillions of different cells, every cell falls into one of two basic categories. Those categories are eukaryotic and prokaryotic. Most eukaryotic cells have a central control structure called a nucleus, which contains the cell's DNA. Organisms composed of eukaryotic cells are called eukaryotes. The term eukaryote comes from the Greek words for good and kernel, referring to the nucleus. A prokaryotic cell does not have a nucleus. Its DNA simply resides in the cytoplasm. An organism consisting of a prokaryotic cell is called a prokaryote. All prokaryotes are one-celled or single-celled organisms and are invisible to the naked eye. So distinguishing a prokaryotic cell from a eukaryotic cell is that eukaryotes are going to have what we call membrane-bound organelles. Prokaryotes do not. Also, I may refer to eukaryotes as euks, and I may refer to prokaryotes as prokes. The first cells on Earth were prokaryotes, so that makes their appearance on Earth about 3.5 to 3.8 billion years ago. The term prokaryote comes from the Greek words for before and kernel, referring to the evolutionary origin of these cells before the eukaryotes. For a long time, that is 1.5 billion years, the prokaryotes had the planet all to themselves. Two groups of prokaryotes exist. They are called bacteria and archaea. All other organisms are eukaryotes. The largest group of prokaryotes is bacteria. Bacteria are probably familiar to you for their ability to cause disease such as pneumonia, to spoil food, and to decompose plant material in compost. They also play important and beneficial roles in human nutrition and health. Less familiar to you may be the archaea. These microorganisms inhabit some of the harshest environments on Earth, thriving in extremes of temperature, salinity, and pH. And this cartoon is located on page 78. This is figure 4.3, and it shows an idealized bacterium. So prokaryotes have four basic structural features. One is a plasma membrane that encompasses the cell, and sometimes is simply called the cell membrane. Anything inside the plasma membrane is referred to as intracellular, and everything outside of the plasma membrane is referred to as extracellular. Also, the cytoplasm refers to the cell's contents contained within the plasma membrane minus the nucleus. Of course, bacteria do not have a nucleus. This includes the jelly-like fluid in the cytoskeleton, collectively called the cytosol, and other organelles. Ribosomes are granular bodies where proteins are made. Thousands of them are scattered throughout the cytoplasm. The DNA of a proc is generally found in the center of the cell. We call this region the nucleoid. Each prokaryote has one or more circular loops of linear DNA strands. These small circular loops of DNA strands are called plasmids, and they are found all throughout the prokaryotic cell. Some procs have additional structures, like many have a rigid cell wall that protects and gives shape to the cell. Some have a sticky, sugary capsule as their outermost layer. The sticky outer coat provides protection and enhances the prokaryote's ability to anchor in place when necessary. Also, many procs have a flagellum or multiple flagella. This is a long, thin, rotating whip-like projection of the plasma membrane that moves the cell through the medium in which it lives. Other appendages include pili, which are much thinner, and they are hair-like projections that help prokaryotes attach to surfaces and through which they can exchange DNA with other cells. Although prokes are smaller, evolutionarily older, and structurally simpler than eukes, they are fantastically diverse metabolically. For example, some can fuel their activities in the presence or absence of oxygen, and some can use as an energy source the sulfur in deep-sea hydrothermal vents or hydrogen gas or light from the sun. And that brings us to our take-home message for section 4.2. Every cell on Earth is either a eukaryotic or prokaryotic cell. Single-celled prokaryotes, which have no nucleus, were the first cells on Earth. Prokaryotes include the bacteria and archaea, and as a group are characterized by tremendous metabolic diversity. And that brings us to eukaryotic cells. In the 2 billion years that eukaryotes have been on the Earth, they have evolved into dramatically different and interesting creatures such as platypuses, dolphins, giant sequoias, and Venus flytraps. Not all eukaryotes are multicellular. Many fungi are unicellular, for example, and the protists are a huge group of eukaryotes, which in most cases are single-celled organisms invisible to the naked eye. Nonetheless, because all prokaryotes are single-celled and thus invisible, all the plants and animals that we see around us are eukaryotic organisms. Moving on, eukaryotic cells are about 10,000 times larger than prokaryotic cells in volume. It is easy to distinguish eukaryotic cells from prokaryotic cells under a microscope. Chief among the distinguishing features of eukaryotic cells is the presence of a nucleus. This is a membrane-enclosed structure that contains linear strands of DNA. Additionally, eukaryotic cells usually contain several other specialized structures in their cytoplasm. These structures are called organelles. Many of these structures are enclosed separately by their own lipid membranes. The physical separation of compartments within a eukaryotic cell provides distinct areas in which different chemical reactions can occur at the same time. In the mostly non-compartmentalized interior of a prokaryotic cell, random molecular movements quickly blend all the chemicals throughout the cell. This reduces the ease with which different reactions can occur simultaneously. So the gist of it is that because eukaryotic cells are compartmentalized, it makes metabolism much more efficient in these types of cells. And that brings us to figure 4.6 on page 80 of your book. And this cartoon illustrates a generalized animal cell. And on this slide, this cartoon is in the same figure, 4.6 on page 80 of your book, and this illustrates a generalized plant cell. So because plant cells and animal cells share a common eukaryotic ancestor, they have much in common, but there are some differences. So unlike animal cells, plant cells are going to have a cell wall, and they also have chloroplasts. Moving on, when you compare a complex eukaryotic cell with the structurally simpler prokaryotic cell, it's hard not to wonder about the origin of eukaryotic cells. The endosymbiotic theory provides the best explanation for the presence of the two types of organelles in eukaryotes, which are chloroplasts in plants and algae, and mitochondria in plants and animals. According to the theory of endosymbiosis, two different types of prokaryotic cells worked in close partnerships. For example, some small prokaryotes capable of performing photosynthesis may have come to live inside a larger host prokaryote. The photosynthetic border may have made some of the energy that it captured in photosynthesis available for use by the host. After a long while, the two cells may have become more dependent on one another, forming a symbiotic relationship in which neither cell could live without the other. Eventually, the photosynthetic prokaryote evolved into a chloroplast, which is the organelle in plant and eukaryotic algae cells in which photosynthesis occurs. Similarly, a large host prokaryote may have engulfed a smaller prokaryote that was unusually efficient at converting food and oxygen into easily usable energy. This smaller prokaryote evolved into what we call a mitochondrion, the organelle in plant and animal cells, as well as in protists and fungi, that converts the energy stored in food into a form usable by the cell. Moving on, this cartoon on page 81 of your book illustrates the idea of the role of endosymbiosis in the evolution of eukaryotes and is supported by several observations. And this slide outlines the evidences that support the theory of endosymbiosis. Chloroplasts and mitochondria are similar in size to prokaryotic cells and divide by splitting, or what we call fission, just as prokaryotes do. Chloroplasts and mitochondria have their own ribosomes, similar to those found in bacteria, that allow them to synthesize some of their own proteins. This ability is not found in other organelles, which rely on proteins made by the cell's cytoplasmic ribosomes. Also, chloroplasts and mitochondria have small amounts of circular DNA. Remember, I was talking about plasmids a couple slides ago. This is similar to the circular DNA in prokaryotes and in contrast to the linear DNA strands found in a eukaryote's nucleus. Furthermore, analysis of chloroplasts and mitochondrial DNA has revealed that it is highly related to bacterial DNA, much more closely than it is related to eukaryotic DNA. The best current theory about the origin of other organelles in eukaryotes focuses on a process called invagination. The idea is that the plasma membrane around the cell may have folded in on itself to form the inner compartments, which subsequently became the modified and specialized organelles that we see in our idealized plant cell and animal cell cartoon that we had at figure 4.6, page 80, we were discussing a couple slides ago. And that brings us to our take-home message for section 4.3, and that is eukaryotes are single-celled or multicellular organisms consisting of cells with a nucleus that contains linear strands of genetic material. Most eukaryotic cells have organelles throughout their cytoplasm. These organelles may have originated evolutionarily through endosymbiosis or invagination or both. Moving on, just as our bodies have a covering of skin, every cell of every living thing on Earth is enclosed by a plasma membrane. That is a thin, flexible, two-layered membrane that holds the contents of a cell in place and regulates what enters and leaves the cell. The plasma membrane is filled with pores, outcroppings, channels, and complex molecules floating around within the two layers of the membrane that facilitate its functions. This concept is often referred to as the fluid mosaic model. So because cells are perpetually interacting with their external environment, plasma membranes must perform several critical functions. First, the membrane enables the cell to take in food and nutrients and dispose of waste products. Also, it is a site at which the cell can build and export molecules needed elsewhere in the body. Furthermore, it mediates communication with the external environment and with other cells, and adhesion to other cells or surfaces. Also, like a border control checkpoint, it controls the flow of molecules, including water, into and out of the cell. Moving on, the foundation of all plasma membranes is a layer of tightly packed lipid molecules called phospholipids. Recall from section 3.8 of this book that phospholipids have what appear to be a head and two long tails. The head consists of a molecule of glycerol linked to a molecule containing phosphorus. This head region is said to be polar because the electrons are not shared equally among the atoms, leading to the regions of partial positive and partial negative charge. The two tails of the phospholipid are long chains of carbon and hydrogen atoms. Because the electrons in these bonds are shared equally, the carbon-hydrogen chains are nonpolar. The chemical structure of phospholipids gives them a split personality. Their polar hydrophilic head region mixes easily with water, and their nonpolar hydrophobic tail region does not mix well with water. This split personality makes phospholipids good membrane material. In the cell's plasma membrane, two sheets of phospholipids are arranged so that all the hydrophobic tails are in contact with one another in the center of the membrane, and the hydrophilic heads are in contact with the watery solution outside and inside the cell. This forms a phospholipid bilayer. These phospholipids are not locked in place. They just float around their side of the bilayer. This resembles similarly charged magnets that push away from each other. The hydrophobic tails in the center of the membranes push away from and avoid coming into contact with the water molecules. Because the center of the bilayer membrane is made up of hydrophobic lipids, the intracellular and extracellular fluids cannot leak across it. In this way, the plasma membrane forms a boundary around the cell's contents. Moving on, the plasma membrane functions with the help of different types of proteins, carbohydrate, and lipid molecules embedded within or attached to the phospholipid bilayer. Because many of these molecules float around within the phospholipid bilayer, held in a proper orientation and prevented from popping out by hydrophobic and hydrophilic forces, the plasma membrane is often described as a fluid mosaic. For every 50 to 100 phospholipids in the membrane, there is one protein molecule. Some of these proteins, called transmembrane proteins, penetrate right through the lipid bilayer from one side to the other. Others, called surface proteins, reside primarily on the inner or outer surface of the membrane. There are several primary types of membrane proteins, each of which has a different function. So this cartoon is on page 84. This is figure 4.10. And we see a cutaway of the fluid mosaic model, and we see the various types of proteins embedded within this membrane or attached to its surface. So let's go through these. First, we'll talk about the receptor proteins, which are either surface or transmembrane proteins that bind to chemicals in the cell's external environment. In doing so, receptor proteins can attach a cell to the extracellular matrix or convey information from the outside to the inside of the cell. Cells in the heart, for example, have receptor proteins within their membrane that bind to adrenaline. This is a chemical released into the bloodstream in times of extreme stress or fright. When adrenaline binds to these heart cells, the cells increase the heart's rate of contraction to pump blood through the body more quickly. Also, we see recognition proteins, which are either surface or transmembrane proteins that give each cell a fingerprint that makes it possible for the body's immune system to distinguish body cells from foreign invaders. Carbohydrates also play a role in recognition. On the outside of the cell membrane, short glycoproteins serve as part of the membrane's fingerprint and help cells bind or adhere to other cells or molecules within the extracellular matrix. Transport proteins are transmembrane proteins that help polar or charged substances pass through the plasma membrane. Membrane enzymes are either surface or transmembrane proteins that accelerate chemical reactions on the plasma membrane surface. The lipid cholesterol can be incorporated in a cell's plasma membrane. Cholesterol helps the membrane maintain its flexibility, preventing the membrane from becoming too fluid or floppy at moderate temperatures and acting as a sort of antifreeze, preventing the membrane from becoming too rigid at freezing temperatures. The membranes of some cells are about 25% cholesterol. Other plasma membranes, such as those of most bacteria and plants, do not contain any cholesterol. And that brings us to our take-home message for section 4.4. Every cell of every living organism is enclosed by a plasma membrane, a two-layered membrane that holds the containers of a cell in place and regulates what enters and leaves the cell. A fluid mosaic of proteins, lipids, and carbohydrates embedded in the plasma membrane helps it perform its functions. Moving on. How important is the plasma membrane of cells in the health of an organism? The most common fatal inherited disease in the United States is cystic fibrosis, which is directly related to the cell's plasma membrane. This disease occurs when an individual inherits from both parents incorrect genetic instructions for producing one type of transmembrane protein, the protein that controls the flow of chloride ions into and out of cells, found primarily in the lungs and digestive tract. These genetic instructions, which can be defective in more than 1,000 different ways, cause malfunction of chloride passageways and thus a gradual accumulation of chloride ions within cells, poisoning them. In nearly all cases of cystic fibrosis, an improper salt balance in the cells occurs. As a consequence, one way to test for cystic fibrosis is to measure the concentration of salt in sweat. Abnormally high concentrations indicate that a person may have this disease. In addition, people with CF experience a buildup of thick, sticky mucus, particularly in the lungs. Normal mucus helps protect the lungs by trapping dust and bacteria. The mucus is then moved out of the lungs, helped along by coughing. But someone with cystic fibrosis cannot move the thick and sticky mucus out of the lungs, so it collects there, where it impairs lung function and increases the risk of bacterial infection. With careful treatment, the life expectancy of someone with CF can be 35 to 40 years or longer. Faulty membranes also play a role in many other diseases, including heart disease, diabetes, and hormonal disorders. In most cases, the improperly functioning membrane component is a receptor protein or a transport protein. Moving on, one product of pharmaceutical research on altering cell membrane functioning is a group of drugs called beta blockers, which are extremely effective at reducing the symptoms of anxiety. Here's how beta blockers work. In stressful situations, your adrenal glands pump out adrenaline. Many cells in your body, particularly the cells of the heart, have receptor proteins called beta-androgenic receptors, or these receptors are sometimes called beta receptors. These receptors are on their plasma membranes and they can bind to adrenaline. When adrenaline binds to beta receptors, it promptly causes your heart to beat faster and more forcefully, increasing your blood pressure. This reaction is useful in a short-term fight or flight situation, but the increased pressure can damage blood vessels over the long run. Depending on its severity, this reaction can also be problematic if you are giving a presentation or taking a test, or if you're in any other anxiety-producing situation. The chemicals in a beta blocker pill travel throughout the body until they encounter the beta receptors. They bind to these receptors, hold on, and block the adrenaline from doing its job. This outcome slows the heart rate. It also causes a reduction in blood pressure, and it can bring greater relief to individuals suffering from the sweating and trembling associated with anxiety. And that brings us to our take-home message for section 4.5. Normal cell functioning can be disrupted when cell membranes do not function properly, but intentional disruption of normal cell membrane function can have beneficial therapeutic effects, such as in the treatment of high blood pressure and anxiety. Moving on, Every cell in your body has a quote-unquote fingerprint made from a variety of molecules located on the outer surface of the cell membrane. Some fingerprint molecules differ from cell to cell depending on the specific function of the cell, whereas others are common to all of your cells. These molecules tell your immune system, quote-unquote, I belong here. Cells with an improper fingerprint are recognized as foreign and are attacked by your body's defenses. Throughout our evolutionary history, this system has helped humans' bodies fight infection. In some cases, however, this vigilance is a problem, like a car alarm that goes off when you don't want it to. For instance, suppose you receive an organ transplant. Even if the donor is a close relative, the molecular fingerprint on the cells of the donated liver is not identical to your own. Your body sees the new organ as a foreign object and fights against it. To prevent organ rejection, doctors must administer drugs that suppress your immune system. Immune suppression helps you tolerate the new organ, but leaves you without some of the defenses needed to fight off other foreign invaders, such as bacteria that may cause infection. The AIDS-causing virus, HIV, uses a molecular fingerprint to infect an individual's cells. These molecular markers, belonging to a group of identifying markers called clusters of differentiation, abbreviated as CD markers, having names such as CD1, CD2, and CD3, these marker molecules are proteins embedded in the plasma membrane. They enable the cell to bind to outside molecules and sometimes transport them into the cell. HIV targets the CD4 marker in conjunction with another receptor. If the virus locates a cell with a CD4 marker, it can infect the cell. But CD4 markers never occur on the surface of skin cells, so casual contact, such as touching, is very unlikely to transmit the virus. Even if millions of HIV particles are present on a person's hands, they just can't gain access to any of the other person's surface cells. The most common ways for HIV to be transferred from one individual to another are through blood, semen, vaginal fluid, or breast milk. Particles of the virus, as well as cells infected by the virus, are present in these fluids. Consequently, the chief routes of transmission are from an infected mother to her child in breast milk, through the use of contaminated needles, and through unprotected sexual intercourse. Because an open sore or cut might expose some of the cells in your bloodstream to the outside world, however, it is not impossible for casual transmission of HIV to occur. And that brings us to our take-home message for section 4.6. Every cell in your body has a quote-unquote fingerprint made from a variety of molecules on the outside-facing surface of the cell membrane. This molecular fingerprint is key to the function of your immune system. Moving on, so far we have examined the cell as if it were a free-living and independent entity. In reality, the majority of cells in any multicellular organism are connected to other cells. In this section, we examine three primary types of connections between animal cells. One is the tight junction, two is desmosomes, and three is the gap junction. Tight junctions form continuous, watertight seals around cells and anchor cells in place, much like the caulking around a tub or sink that keeps the water from leaking into the surrounding walls. Tight junctions prevent fluid flow between cells. Tight junctions are particularly important in the small intestine. Desmosomes are like small regions of Velcro that fasten cells together into strong sheets. Desmosomes occur at irregular intervals and hold cells together but are not watertight, allowing fluid to pass around them. Desmosomes and similar junctions are found in much of the tissue that lines the cavities of animals' bodies. They also are found in muscle tissue, holding fibers together. And gap junctions are pores surrounded by special proteins that form open channels between two cells. Functioning like a secret passageway, these junctions are an important mechanism for cell-to-cell communication, even if it involves molecules that would be too large to simply pass through adjacent cells' membranes. In the heart, for example, the electrical signal that tells muscle cells to contract is passed from cell to cell through gap junctions. Compared with normal cells, cancer cells have fewer gap junctions. Research suggests that the resulting reduction in intercellular communication among cancer cells may be important in the formation of masses of cells called tumors and the spread of cancer cells throughout the body. Interestingly, treatments that cause an increase in the number of gap junctions tend to reduce tumor growth. And that brings us to our take-home message for section 4.7. In multicellular organisms, most cells are connected to other cells. Tight junctions hold sheets of cells together. Desmosomes allow fluid to pass between neighboring cells, and gap junctions function like passageways, allowing the movement of cytoplasm, molecules, and other signals between cells. Moving on, to function properly, cells must take in food and/or other necessary materials and must move out both metabolic wastes and molecules produced for use elsewhere in the body. When this movement requires energy, the process is called active transport. And when molecular movement occurs spontaneously without the input of energy, the process is called passive transport. There are two types of passive transport: diffusion and osmosis. Diffusion is passive transport in which a particle called a solute is dissolved in a gas or liquid, a solvent, and moves from an area of high solute concentration to an area of lower concentration. A difference in the concentration of solutes in two areas is called a concentration gradient. We say that molecules tend to move quote-unquote down their concentration gradient. As a simple illustration of this idea, add a drop of food coloring into a bowl of water and wait a few minutes. The dye molecules initially cluster together in a very high concentration and so keep bumping into each other. But gradually, they disperse down their concentration gradient until the color is equally spread throughout the bowl. We see this concept illustrated on the cartoon on your slide, and this cartoon is on page 89, figure 4.15. In cells, molecules such as oxygen and carbon dioxide that are small and carry no charge can pass directly through the phospholipid bilayer of the membrane. In a process called simple diffusion, each time you take a breath, for example, there is a higher concentration of oxygen molecules in the air you pull into your lungs than in the blood in your lungs. As a result, oxygen diffuses across the plasma membranes of the lung cells into your bloodstream, where the red blood cells pick up the oxygen and deliver it to where it is needed. Similarly, because CO2 is present at a higher concentration in your bloodstream than in the air in your lungs, it diffuses from your blood into the cells of your lungs and is released to the atmosphere when you exhale. Most molecules, however, can't get through the plasma membranes by simple diffusion. Polar molecules are repelled by the hydrophobic middle region of the phospholipid bilayer. Other molecules may simply just be too big to squeeze through the membrane. These molecules may still be able to diffuse across the membrane down their concentration gradient, but only with the help of a transport protein. This is a process called facilitated diffusion. A transport protein spans the membrane and functions like a revolving door, allowing the movement of molecules in either direction depending on their concentration gradient. Moving on, just as solute molecules will passively diffuse down their concentration gradients, water molecules will move from areas of higher concentration to areas of lower concentration in order to equalize the concentration of water inside and outside of the cell. The diffusion of water across a membrane is a type of passive transport that we call osmosis. As solute molecules move across the membrane, molecules of water to move across the membrane. This equalizes the concentration inside and out of the cell. Although some water can pass through the lipid bilayer, the hydrophobic region severely limits this flow. Most of the rapid movement of water into and out of the cells occurs through water channels called aquaporins, which are transmembrane proteins with hydrophilic channels through which water molecules can pass in single file. Osmosis can have some dramatic effects on cells. As we saw earlier, many molecules just can't move across a cell membrane. Water, however, can move into the cell down the concentration gradient, causing the cell to swell or to shrink. So this is how osmosis works. In a fluid environment, dissolved substances are located inside as well as outside of the cell. The relationship between the concentrations of solutes inside the cell and solutes outside of the cell is referred to as tonicity. If the concentration of solutes outside of the cell is equal to the concentration inside of the cell, the outside solution is what we call isotonic. Water still moves between the solution and the cell, but at the same rate in both directions. So there is no net change in the amount of water inside versus outside of the cell. However, if the concentration of solutes outside the cell is lower than the concentration inside the cell, the outside solution is what we call hypotonic. In this situation, if the cell membrane is not permeable to solutes because the water molecule concentration is greater outside the cell, water will move into the cell and the cell will swell. Thirdly, if the concentration of solutes outside the cell is higher than the concentration inside the cell, the outside solution is said to be hypertonic. In this situation, if the cell membrane is not permeable to the solutes because the water molecule concentration is greater inside the cells, more water will move out of the cell than into it, and in turn, the cell will shrivel. Moving on, you can see osmosis in action in this cartoon. This is figure 4.18 on page 91 of your book. So go to your kitchen and take a stalk of celery and leave it on the counter for a couple of hours. As water evaporates from the cells of the celery stalk, it will shrink and become limp. You can make it crisp again by placing it in a solution of distilled water. Why distilled water? Because it contains very few dissolved molecules. It's a hypotonic solution. The celery stalk cells, however, contain many dissolved molecules such as salt that can't easily pass across the plasma membrane of the celery stalk cells. So, the water molecules move down their concentration gradient from the distilled water into the celery cells. Moving on, would the celery regain its crispness if you placed it in concentrated salt water? No, because there would be more dissolved solute molecules and fewer water molecules in the hypertonic salt solution than inside the celery cells. What little water remained in the celery cells might actually move by osmosis out of the celery, causing it to shrivel even more. It is important to note that the direction of osmosis is determined only by a difference in the total concentration of all the molecules dissolved in the water. It does not matter what the solutes are, only how many molecules of solutes there are. To determine which way the water molecules will move, you need to determine the total amount of quote-unquote dissolved stuff on either side of the membrane. The water will move toward the side with the greater concentration of dissolved stuff. Because of osmosis, even small increases in the salinity of lakes can have disastrous consequences for the aquatic organisms living there, from fish to bacteria. And that brings us to the take-home message for section 4.8. Passive transport is the movement of molecules and other substances across cell membranes without the input of energy. This generally takes place as molecules move down their concentration gradient. The diffusion of water across a membrane from an area with a lower concentration of solutes to an area with a higher concentration of solutes is a type of passive transport that we call osmosis. And that brings us to active transport. Active transport into and out of cells may be necessary if molecules or ions are very large or are being moved against their concentration gradient. In all active transport, proteins embedded in the membrane act like motorized revolving doors, pushing molecules across cell membranes regardless of the concentration of those molecules on either side of the membrane. Moving on, the two distinct types of active transport, primary and secondary, differ only in the source of the fuel that keeps the revolving doors spinning. The process of digestion provides an example of primary active transport, the type of active transport that occurs when the energy from ATP is used to fuel the transport of molecules. To help break down food into more digestible bits, the cells lining your stomach create an acidic environment by pumping large numbers of hydrogen ions into the stomach contents against their concentration gradient. All of this hydrogen ion pumping increases your ability to digest food, but requires the use of the high-energy molecule ATP because protons would not normally flow into a region against their concentration gradient. ATP stands for adenosine triphosphate, and this is a very important molecule to your body because it is an energy molecule, and we will discuss this molecule more in subsequent chapters. Many transport proteins use an indirect method of fueling their activities rather than using energy released directly from ATP. In the process of secondary active transport, the transport protein simultaneously moves one molecule against its concentration gradient while letting a second type of molecule flow down its concentration gradient. Although no ATP is used directly in this process, at some other time and in some other location, energy from ATP was used to pump the second type of molecule against its concentration gradient. This process is like using energy to pump water to the top of a high water tower. Later, the water can be allowed to run out of the tower over a water wheel, which can in turn power a process such as grinding wheat into flour. Our bodies frequently use the energy from one reaction that occurs spontaneously to fuel another reaction that requires energy. And that brings us to our take-home message for section 4.9. Active transport is necessary if the molecules to be moved are very large or if they are to be moved against their concentration gradient. Movement of molecules across a membrane requires energy. In such cases, proteins embedded in the plasma membrane pump the molecules inside or outside the cell. Moving on, many substances are just too big to get into or out of the cell by passive or active transport. To absorb large particles such as bacterial invaders, cells engulf them with their plasma membrane in a process called endocytosis. To export large particles, such as digestive enzymes manufactured for use elsewhere in the body, cells often use the process of exocytosis. There are three types of endocytosis. They are phagocytosis, pinocytosis, and receptor-mediated endocytosis. In all three types of endocytosis, the plasma membrane oozes around an object outside the cell, surrounding the object, and forms a little pocket-like sac called a vesicle. The plasma membrane then pinches off the vesicle so that the vesicle is inside the cell but separated from the rest of the cell contents. Relatively large particles are engulfed in a process called phagocytosis. Amoebas and other unicellular protists, as well as white blood cells, use phagocytosis to consume entire organisms for defense or food, whereas phagocytosis comes from the Greek words for eat and container, the term pinocytosis comes from the Greek words drink and container and describes the process of cells taking in dissolved particles and liquids. The two processes are largely the same, except that vesicles formed during pinocytosis are generally much smaller than those formed during phagocytosis. In receptor-mediated endocytosis, receptor molecules on the surface of a cell recognize and bind to one specific type of molecule. When the appropriate molecule binds to each of the receptor proteins, the membrane begins to fold inward, first forming a large pit, and then eventually completely engulfing the molecules, which are still attached to their receptors. For example, in the bloodstream, most cholesterol is in the form of globules coated by phospholipids, called low-density lipoproteins, or LDLs. Receptor proteins built into the liver cell plasma membranes recognize and bind the proteins embedded within the globules' phospholipid coat. This enables those cells to consume the globule, break down the cholesterol, and use it to make a variety of useful molecules such as estrogen and testosterone. Circulating cholesterol collects on the walls of arteries, reducing blood flow and causing the artery to harden. Too much circulating cholesterol in LDL molecules can lead to cardiovascular disease and death. Individuals lucky enough to have large numbers of LDL receptors on the plasma membranes of their liver cells have significantly lower risk of cardiovascular disease. Conversely, some individuals have the misfortune of inheriting genes that code for faulty liver cell membranes that have few LDL receptors. This is a disorder called familial hypercholesterolemia. In the extreme case where an individual is born with no LDL receptors, circulating cholesterol accumulates in the artery so rapidly that cardiovascular disease begins to develop even before puberty, and death from a heart attack can occur before the age of 30. That brings us to exocytosis. To shuttle molecules out of the cell, particles within the cell are first enclosed in a vesicle. The vesicle then moves through the cytoplasm to the plasma membrane. Once the vesicle is at the plasma membrane, it then will merge with the plasma membrane, and the material in the vesicle is expelled from the cell. The hormone insulin, for example, is transported out of pancreatic cells in this manner. Exocytosis is not restricted to large molecules. In the brain and other parts of the nervous system, for example, communication between cells occurs as one cell releases large numbers of very small molecules called neurotransmitters by exocytosis. And this is figure 4.20 on page 93 of your book. And you can see through phagocytosis, cells engulf particles or entire organisms for food or for defense. And on this next slide, we see the cartoon also on page 93, but this is figure 4.21, and we see receptor proteins on liver cells bind cholesterol, helping remove it from the bloodstream. And this is on page 94. This is figure 4.22. This is a comparison of a healthy artery and an artery choked by the buildup of cholesterol. And this next slide shows us figure 4.23, which is also on page 94. And this cartoon outlines exocytosis. Exocytosis moves molecules out of the cell. This is how insulin is exported from cells where it is synthesized. And that brings us to our take-home message for section 4.10. When materials cannot get into a cell by diffusion or through a pump, cells can engulf the molecules or particles with their plasma membrane in a process called endocytosis. The plasma membrane moves to surround the molecule and forms a vesicle that pinches off from the inside of the cell. Similarly, molecules can be moved out of the cell by exocytosis, in which a vesicle inside the cell fuses with the plasma membrane and dumps its contents outside of the cell. Moving on, the nucleus is the largest and most prominent organelle in most eukaryotic cells. In fact, The nucleus is generally larger than any prokaryotic cell. Three important structural components stand out in the nucleus. First is the nuclear membrane, sometimes called the nuclear envelope, which surrounds the nucleus and separates it from other parts of the cytoplasm. Unlike most plasma membranes, however, the nuclear membrane consists of two bilayers, one on top of the other, much like the double bagging of groceries at the market. The nuclear membrane is a very leaky bag, though. It is perforated with tiny pores that enable large molecules to pass between the nucleus and the cytosol. Made from multiple proteins embedded in the phospholipid membranes and spanning both bilayers of the nuclear envelope, these pores also permit free diffusion of small molecules such as water, sugar, and ions. And the cartoon on this slide is figure 4.25 on page 96. The nuclear pore protein complexes regulate the transport of many molecules across the nuclear envelope in both directions. Two types of transport are particularly important. The nuclear membrane allows proteins that interact with DNA and catalyze nuclear activities to move from the cytosol into the nucleus. They also facilitate the movement of RNA and RNA protein complexes from the nucleus to the surrounding cytosol. The second prominent structure in the nucleus is chromatin, a mass of long, thin fibers consisting of DNA with some proteins attached that keep the DNA from getting impossibly tangled. Most of the time, as the DNA directs cellular activities, the chromatin resembles a plate of spaghetti. When it's time for the cell to divide, the chromatin coils and the threads become shorter and thicker until they become visible as chromosomes, the compacted linear DNA molecules that carry hereditary information. And we'll talk more about cell division in a subsequent chapter. The third structure in the nucleus is the nucleolus, an area near the center of the nucleus where subunits of ribosomes, a critical part of the cellular machinery, are assembled. Ribosomes are like little factories in which information is stored in DNA and it is used to construct proteins. The ribosomes are built in the nucleolus but pass through the nuclear pores into the cytosol before starting their protein production work. And that brings us to our take-home message for section 4.11, and that is the nucleus is usually the largest and most prominent organelle in the eukaryotic cell. It directs most cellular activities by controlling which molecules are produced and in what quantity. The nucleus is the storehouse for hereditary information. Moving on, if you imagine yourself inside a cell the size of a big lecture hall, it might come as a surprise that you can barely see the big rig of a nucleus parked in the front rows. Visibility is almost zero, not only because the room is filled with jelly-like cytosol, but also because there is a dense web of thick and thin, straight and branched ropes, strings, and scaffolding running every which way throughout this room. The inner scaffolding of the cell, which is made from proteins, is the cytoskeleton. It has three chief purposes. First, it gives animal cells shape and support, making red blood cells look like little round donuts, and giving neurons their very long thread-like appearance. Plant cells, on the other hand, are shaped primarily because of their cell wall, but they also have a cytoskeleton. Secondly, the cytoskeleton controls the intracellular traffic, serving as a series of tracks on which a variety of organelles and molecules are guided across and around the inside of the cell. Third, because the elaborate scaffolding of the cytoskeleton is dynamic and can generate force, it gives all cells some ability to control their movement. Three types of protein fibers make up the cytoskeleton. Microtubules, the thickest, are linear fibers made from repeating units of protein and look like rigid hollow tubes. Like intracellular conveyor belts, they are the tracks to which molecules and organelles within the cell can become attached and moved along. Microtubules also help to pull chromosomes apart during cell division. Continuously built, dismantled, and rebuilt, microtubules rarely last more than 10 minutes in a cell. Intermediate filaments, a second type of cytoskeleton fiber, are durable, rope-like systems of numerous different overlapping proteins. They give cells great strength. Microfilaments are the thinnest elements in the cytoskeleton. These long, solid, rod-like fibers help generate forces, including those important in cell contraction and cell division. Moving on, a couple of microtubule-based structures are sometimes present in cells and can help move the cell through its environment, or in stationary cells can help move the environment past the cell. Cilia are short projections often found in large numbers on a single cell. Cilia beat swiftly, often in unison, and in ways that resemble the blades of grass in a field blowing in the wind. Cilia can move fluid along and past the cell. This movement can accomplish many important tasks, including sweeping the airways to our lungs to clear them of debris such as dust. Centrioles found in animal cells are made from bundles of microtubules and play a role in cell division. Flagella are much longer than cilia. They occur in many prokaryotes and single-celled eukaryotes, although the flagella of prokaryotes differ structurally from those of eukaryotes. While many algae and plants have cells with one or more flagella, in animals, cells with one or more flagella are rare. One of these types of cells, however, has a critical role in every animal species. Sperm cells with a flagella for a tail are among the most mobile of all animal cells. Some spermicidal birth control methods prevent conception by disabling the flagellum and immobilizing the sperm cells. And that brings us to our take-home message for section 4.12. The cytoskeleton comprises microtubules, intermediate filaments, and microfilaments. These structures give animal cells their shape and support, allow some cells to control their movement, and serve as a series of tracks on which organelles and molecules can move inside of the cell. Moving on, cars generally run on a single type of fuel, which is exactly the same fuel every time. With human bodies, it's a different story. During some meals, we fill our bodies with meat and potatoes, at other times fruit or bread or vegetables, popcorn or gummy bears. Yet our bodies can use the energy in these various foods to power all the reactions that make it possible for us to breathe, move, and think. The mitochondria are the organelles that make this possible. Mitochondria are all-purpose energy converters that are present in nearly all plant cells, animal cells, and every other eukaryotic cell. Our mitochondria convert energy contained in the chemical bonds of carbohydrates, fats, and proteins into carbon dioxide, water, and ATP. Cells use high-energy ATP molecules to fuel their functions and activities. Because this energy conversion requires a significant amount of oxygen, mitochondria consume most of the oxygen used by each cell. In humans, for example, our mitochondria consume as much as 80% of the oxygen we breathe. Mitochondria give a significant return on this investment by producing about 90% of the energy our cells need to function. Moving on, cells that are not very metabolically active, such as some fat storage cells in humans, have very few mitochondria. By contrast, cells that have large energy requirements, such as muscle, liver, and sperm cells in animals and fast-growing root cells in plants, are packed densely with mitochondria. Some of these cells have as many as 2,500 mitochondria. To visualize the structure of a mitochondrion, imagine a plastic sandwich bag. Now take another bigger plastic bag and stuff it inside the plastic sandwich bag. There is a smooth outer membrane and a scrunched up inner membrane. This construction forms two separate compartments within the mitochondrion. A region outside the inner bag called the inner membrane space and another region called the mitochondrial matrix inside the inner bag. This bag within a bag structure has important implications for energy conversion. The large, heavily folded inner membrane also provides a huge amount of surface area on which to conduct chemical reactions. Moving on, as we learned earlier in our discussion of endosymbiosis, billions of years ago, mitochondria may very well have existed as a separate, single-celled, bacteria-like organism. Perhaps the strongest evidence for this is that mitochondria have their own DNA. Anywhere from 2 to 10 copies of its own little ring-shaped DNA are mixed in among the approximately 3,000 proteins in each mitochondrion. This DNA carries the instructions for making 13 mitochondrial proteins essential for metabolism and energy production. We're generally taught that our mothers and fathers contribute equally to our genetic composition, but this isn't quite true. The mitochondria in every one of your cells and the DNA that comes with them come from the mitochondria that were initially present in your mother's egg, which, when fertilized by your father's sperm, developed into you. In other words, all of your mitochondria are descended from your mother's mitochondria. The tiny sperm contributes DNA, but no cytoplasm and hence no mitochondria. Consequently, mitochondrial DNA, or mDNA, is something that we inherit exclusively from our mothers. This is not only true in humans, but also in most multicellular eukaryotes. Given the central role of mitochondria in converting energy and food molecules into a form that is suitable by cells, you won't be surprised to learn that mitochondrial malfunctions can have serious consequences. Recent research has focused on possible links between defective mitochondria and diseases characterized by fatigue and muscle pain. It seems, for instance, that many cases of exercise intolerance, extreme fatigue, or cramps after only slight exertion may be related to defective mitochondrial DNA. And that brings us to our take-home message for section 4.13. In mitochondria, which are found in nearly all eukaryotic cells, the energy contained in the chemical bonds of carbohydrate, fat, and protein molecules is converted into carbon dioxide, water, and ATP, the energy source for all cellular functions and activities. Mitochondria may have evolutionary origins as symbiotic bacteria living inside other cells. Moving on, garbage. What does a cell do with all the garbage it generates? Mitochondria, for example, wear out after about 10 days of intense activity. White blood cells constantly track down and consume bacterial invaders, which they then have to dispose of. Similarly, the ongoing reactions of cellular metabolism produce many waste macromolecules that cells must digest and recycle. To deal with this garbage, many eukaryotic cells maintain hundreds of versatile floating quote-unquote garbage disposals that we call lysosomes. Lysosomes are round, membrane-enclosed, acid-filled vesicles that dispose of garbage. They are filled with about 50 different digestive enzymes and a superacidic fluid, a corrosive broth so powerful that a burst of lysosome would rapidly kill the cell by digesting many of its component parts. The broad spectrum of enzymes in the lysosome dismantle macromolecules that are no longer needed by the cell or are generated as byproducts of cellular metabolism. When a cell consumes a particle of food or even an invading bacterium via phagocytosis, the cell directs the material to lysosomes for dismantling. After this dismantling, the cell releases most of the component parts of molecules, such as amino acids, from proteins into its cytoplasm, where they can be used by the cell as raw materials. Some immune system cells tend to have particularly large numbers of lysosomes, most likely because of their great need for disposing of the byproducts of disease-causing bacteria. As 50 different enzymes are necessary for lysosomes to function, sometimes problems may occur. In a common genetic disorder called Tay-Sachs disease, a child inherits an inability to produce a critical lipid-digesting enzyme. Even though the lysosomes cannot digest certain lipids, the cells continue to send lipids to the lysosomes where they accumulate undigested. The lysosome swells until it bursts and digests the whole cell or chokes the cell to death. This process occurs in large numbers of cells within the first few years of life and eventually leads to a child's death. Although the matter has long been debated among biologists, it seems that plant cells also contain lysosomes, compartments with similar digestive broths that carry out the same digestive processes as in animal cells. And that brings us to our take-home message for section 4.15, and that is lysosomes are membrane-enclosed organelles filled with about 50 different digestive enzymes that enable a cell to dismantle macromolecules, including disease-causing bacteria. Moving on, the information for how to construct the molecules essential to a cell's functioning is stored in the DNA found in the cell's nucleus. The energy used to construct these molecules and to run cellular functions comes primarily from the mitochondria. The actual production and modification of biological molecules, however, occurs in a system of organelles called the endomembrane system. This mass of interrelated membranes spreads out from and surrounds the nucleus, forming chambers within the cell that contain their own mixtures of chemicals. The endomembrane system occupies as much as 1/5 of the cell's volume and is responsible for many of the cell's fundamental functions. Moving on, rough ER, perhaps the organelle with the most cumbersome name, the rough endoplasmic reticulum, or rough ER, as I already said, is a large series of interconnected flattened sacs. These sacs are connected directly to the nuclear envelope and, in most eukaryotic cells, almost completely surround the nucleus. The rough surface is studded with ribosomes, the cell's protein-making machines. Cells with high rates of protein production generally have large numbers of ribosomes. The primary function of the rough ER is to fold and package proteins for shipments to other locations in the endomembrane system, to the cell surface, or to the outside of the cell. Proteins destined for use within the cell are generally produced on free-floating ribosomes in the cytoplasm. Poisonous frogs, for example, package their poison in the rough ER of poison-producing cells before transporting it to the poison glands in their skin. This brings us to the smooth ER, or the smooth endoplasmic reticulum, and this is also part of the endomembrane network, but it is smooth because it has no ribosomes bound to it. Although connected to the rough ER, it is located farther from the nucleus. The smooth ER sometimes looks like a collection of branched tubes. Smooth ER is not extensively involved in folding or packaging proteins. Instead, it takes part in the synthesis of lipids such as fatty acids, phospholipids, and steroids, as well as carbohydrates. The exact lipids produced vary throughout the organism and across species. The smooth ER of mammalian ovaries and testes, for example, produces the hormones estrogen and testosterone. The smooth ER of liver and fat cells produce other lipids. Following the same packaging process that occurs for proteins in the rough ER, lipids produced by the smooth ER are packaged in transport vesicles and then sent to other parts of the cell or to the plasma membrane for export. A critical responsibility of smooth ER, particularly in human cells, is to help protect us from the many dangerous molecules that get into our bodies. Detoxifying enzymes in smooth ER reduce the harmfulness of alcohol, antibiotics, barbiturates, amphetamines, and other stimulants that we may consume, along with many toxic metabolic waste products formed in our body. And just as a bodybuilder's muscles get bigger in response to weightlifting, smooth ER proliferates in cells exposed to large amounts of particular drugs. Moving on, chronic exposure to many drugs, from antibiotics to heroin, can induce a proliferation of smooth ER and detoxification enzymes, particularly in the liver. This proliferation increases tolerance to drugs, necessitating higher doses to achieve the same effect. The cell's increased detoxification capacity often enables them to better detoxify other compounds as well. An individual who has been exposed to large amounts of certain drugs, for example, may end up responding less well to antibiotics or anesthesia. Other cells that are packed with ER, both rough and smooth, include blood cells called plasma cells, which produce immune system proteins, and pancreas cells that secrete large amounts of digestive enzymes. Moving farther outward from the nucleus, we encounter another organelle within the endomembrane system. The Golgi apparatus is a flattened stack of membranes, each called a Golgi body, which are not connected to the ER. The Golgi apparatus processes molecules synthesized in the cell, primarily proteins and lipids, and packages those destined for use elsewhere. It is also a site of carbohydrate synthesis, including complex polysaccharides found in many plasma membranes. To reach the Golgi apparatus, transport vesicles bud from the ER and move through the cytosol. The vesicles fuse with the Golgi apparatus membrane and dump their contents into the Golgi body. As the molecules pass through about 4 successive Golgi body chambers, enzymes make slight modifications to the molecules, such as adding or removing phosphate groups or sugars. The processing that occurs in the Golgi apparatus often involves tagging molecules, much like adding a postal address to direct them to some other part of the organism. After processing, the molecules bud off from the Golgi apparatus in a vesicle. If the molecules are destined for delivery and use elsewhere in the body, the transport vesicle eventually fuses with the cell's plasma membrane and dumps the molecules into the bloodstream via exocytosis. And moving on to this figure 4.35, which is on page 105 of your book, this cartoon shows how the various parts of the endomembrane system work to produce, modify, and package molecules within a cell. First, a transport vesicle buds from the smooth or rough ER. Second, a transport vesicle fuses with the Golgi apparatus, dumping the contents inside. Thirdly, the Golgi apparatus modifies the molecules as they move through its successive chambers. Fourth, modified molecules bud off from the Golgi apparatus in a transport vesicle. And finally, the vesicle may fuse with the plasma membrane, dumping contents outside the cell for delivery elsewhere in the organism. And that brings us to our take-home message for section 4.16. The production and modification of molecules occurs in a system of organelles called the endomembrane system. In the rough endoplasmic reticulum, proteins are folded and packaged. In the smooth endoplasmic reticulum, lipids and carbohydrates are synthesized and various chemicals are detoxified. The Golgi apparatus processes molecules and packages those destined for use elsewhere. Moving on, until this point, we've discussed organelles and structures that are common to both plants and animals. Now we'll look at several structures that are not found in all eukaryotic cells. Because plants are rooted into the ground, unable to move, they have very special needs beyond those of animals. In particular, they can't outrun predators or outmaneuver their competitors to get more food. If they need more light, they have a few options beyond simply growing larger or taller, and to resist plant-eating animals, they must simply reduce their edibility. One structure that helps the plants achieve those goals is the cell wall, which surrounds the plasma membrane. The plant cell wall is made largely from long fibers of a polysaccharide called cellulose. Animals do not have cell walls, but some non-plants such as archaea, bacteria, protists, and fungi do. Their cell walls are made of a polysaccharide other than cellulose. The cell wall is nearly 100 times thicker than the plasma membrane. The tremendous strength it confers enables some plants to grow several hundred feet tall with exposure to less shaded light. And the wall provides some protection from feeding insects and other animals. Cell walls can also help make plants more watertight, an important feature in an organism that cannot move out of the hot sun to reduce water loss through evaporation. Yet, the cell wall is porous, allowing water and solutes to reach the plasma membrane. Additionally, the cells of most plants have anywhere from 1,000 to 100,000 microscopic tube-like chemicals called plasmodesmata, which connect the cell to each other, enable communication and transport between them. In fact, because so many of the cells are connected to one another, sharing cytoplasm and other molecules, some biologists have wondered whether we should consider a plant as just one big cell. And that brings us to our take-home message for section 4.17. The cell wall in plants confers tremendous structural strength to plant cells, gives plants increased resistance to water loss, and provides some protection from insects and other animals that might eat the plant. Plasmodesmata connect the plant cells and enable communication and transport among them. Moving on, if you look at a mature plant cell through a microscope, one organelle, the central vacuole, usually stands out because it is so huge and appears empty. But it is not empty. Surrounded by a membrane filled with fluid and occupying from 50 to 90% of the plant cell's interior space, the central vacuole can play an important role in 5 different areas of plant life. One is nutrient storage. The vacuole stores hundreds of dissolved substances, including amino acids, sugars, and ions. Waste management. The vacuole retains waste products and degrades them with digestive enzymes, much like the lysosome in animal cells. Thirdly, predator deterrence. The poisonous, nasty-tasting materials that accumulate inside the vacuoles of some plants act as powerful deterrents to animals that might try to eat parts of the plant. Fourthly, sexual reproduction. The vacuole may contain pigments that give some flowers the colors that attract pollinators. Pollinators can be birds or insects that help the plant reproduce by transferring pollen. Finally, physical support. High concentrations of dissolved substances in the vacuole can cause water to rush into the cell via osmosis. The increased fluid pressure inside the vacuole can cause the cell to enlarge and push against the cell wall. This process is responsible for the pressure that we call turgor pressure that allows stems, flowers, and other plant parts to stand upright. The ability of non-woody plants to stand upright is due primarily to turgor pressure. Wilting is the result of a loss of turgor pressure. Vacuoles are also found in some other eukaryotes, including some protists, fungi, and animals, but they tend to be particularly prominent in plant cells. And that brings us to our take-home message for section 4.18. Vacuoles are storage spaces and play roles in nutrition, waste management, predator deterrence, reproduction, and physical support. Vacuoles can occupy most of the interior space of the cell. Vacuoles are also present in some other eukaryotic species, but are usually less prominent in these organisms. Moving on, it would be hard to choose the most important organelle, but if we had to, the chloroplast would be a top contender. The chloroplast is an organelle found in all plant cells and eukaryotic algae, and it is the site of photosynthesis. Photosynthesis is the conversion of light energy into the chemical energy of food molecules, which produces oxygen as a byproduct. Because all photosynthesis in plants and algae takes place in the chloroplast, these organelles are directly or indirectly responsible for everything we eat and for the oxygen that we breathe. In a green leaf, each cell has about 40 to 50 chloroplasts. This means that there are about 500,000 chloroplasts per square millimeter of leaf surface. That's more than 200 million chloroplasts in an area the size of a small postage stamp. Chloroplasts are oval, somewhat flattened organelles. The outside of the chloroplast is encircled by two distinct layers of membranes, similar to the structure of mitochondria. Inside the compartment is a fluid called the stroma, which contains some DNA and much of the protein-making machinery. With a simple light microscope, little spots of green are visible within the chloroplasts. Up close, these spots look like stacks of pancakes. Each stack consists of numerous interconnected little flattened sacs called thylakoids. The light-collecting process for photosynthesis occurs on the membranes of the thylakoids. A peculiar feature of chloroplasts mentioned in section 4.3 is that they resemble photosynthetic bacteria, particularly with their circular DNA, which contains many of the genes essential for photosynthesis. Also, the dual outer membrane of the chloroplast is consistent with the idea that long ago a predatory cell engulfed a photosynthetic bacterium, enveloping it with its plasma membrane in endocytosis, and that chloroplasts are derived from these bacteria. And that brings us to our take-home message for section 4.19. The chloroplast is the organelle of plants and algae that is the site of photosynthesis. That is the conversion of light energy into chemical energy, which produces oxygen as a byproduct. Chloroplasts may originally have been bacteria that were engulfed by a predatory cell by endosymbiosis. And in this figure, we see a summary or review of the major cellular structures and their functions. This can be found on page 108 of your book, and this is figure 4.39. The nucleus directs cellular activity and stores hereditary information. The cytoskeleton provides structural shape and support and enables cellular movement. The mitochondria harvests energy for cellular functions. Lysosomes digest and recycle cellular waste products and consumed material. The rough ER modifies proteins that will be shipped elsewhere in the organism. The smooth ER synthesizes lipids and detoxifies molecules. The Golgi apparatus processes and packages proteins, lipids, and other molecules. The cell wall provides structural strength, protection, and increased resistance to water loss. The vacuole stores nutrients, degrades waste, contains pigments, and provides structural support. And the chloroplast performs photosynthesis.